Okay, then. Yeah, so I'm really sorry that that I was in the wrong room. Yeah, but welcome to the second session of our symposium on evolutionary applications. And our first speaker in this session is Madhavi Colton. She's currently the Vice President and Executive Director of Audubon California. This is a non-profit organization which is dedicated to bird and wildlife conservation. But before that, she has worked for a really long time at the Coral Reef Alliance. And this is what she's going to talk about today. And I'm really, really happy that you're here.
Thank you. And hello, everybody. I was going to say good morning. It's morning. I'm calling in from California, but I know we've got a variety of time zones here. So welcome, everybody. Let me go ahead and share my screen. Can you see my presentation?
Yes.
Great. Excellent. Well, as Hildegard mentioned, I'm currently at Audubon, California, but presenting work that I did while I was at the Coral Reef Alliance. And my talk today is going to be about evolutionary adaptation on coral reefs, with a real focus on how we turn some of this science into conservation and management actions at a variety of scales. But I want to start by just reminding us all why we should care so deeply about coral reefs. They are truly amazing ecosystems. The coral animals themselves build structures that are big enough to be seen from space. 25% of all marine life relies on coral reefs, even though they only occupy 0.1% of the ocean floor. Coral reefs protect shorelines. A healthy reef can reduce wave height by 70% and wave intensity by 97%. And around the world, they support 500 million people. And they're estimated to be worth at least $375 billion per year in goods and services. And that doesn't even get at their existence value. For many cultures, corals play a critical role in their cultural traditions. For example, in Hawaii, the Hawaiian creation story is that all life started as a coral polyp. And unless you've been hiding under a rock recently, you know that coral reefs are in trouble. We've lost about 14% of the world's coral reefs over this last decade. That's equivalent to losing all of the corals in Australia. And scientists worry that by the middle of this century, the heat waves are going to come so frequently that corals won't have the chance to recover between them. And so all coral reefs will be threatened. So declines on coral reefs are driven by things that happen at both global and local scales. Global climate change and the associated increases in temperatures have negative impacts on reefs. And you can see this photo here from American Samoa. On the left, we have a healthy reef. On the right, that reef is bleached. And so in case you don't know what bleaching is, corals are actually a symbiotic organism. Part of that symbiosis is the coral animal host. And part of it is that inside their tissues, there are algae living inside their tissues called zooxanthellae. Those algae provide food to the coral using photosynthesis. And when the water around a reef gets too warm for too long, that symbiotic relationship breaks down. The corals kick those zooxanthellae out. With the zooxanthellae goes the coral color, and they start to turn bright white. If that event goes on too long, the corals start to starve and then to die. And that process can be quite rapid, as shown here. This was the previous to our current global coral bleaching event, where we have a healthy reef in December. By February of the following year, that reef is bleached, and by August, it is dead. But that's what's happening globally. There's also a bunch of things happening locally that negatively impact reefs as well. We've got sedimentation, overfishing, direct damage to reefs, wastewater discharge into the marine environment, and other water quality issues. And there are some really nasty interactive effects when you get heat waves interacting with some of these local stresses. So this is kind of the result of that in the coral conservation community, all of us really wondering what on earth we can do to protect this critical ecosystem. And as a scientist, of course, I want to base what we should do in the best science about how species can deal with these effects themselves. So I'm going to simplify here 3 types of responses that species can have to rapid environmental change. The first is movement, and there are numerous examples out there of shifts in range, expansions and contractions in range, elevational changes in range distributions. I've provided a couple here from Malin Pinsky's work showing shifts in distribution of a couple of marine animals, but I'm sure that whatever system you're working on, you've got plenty of examples of that as well. Evolutionary adaptation is also a response to climate change, and as I'm sure you all know, this can be quite rapid. One of the clearest examples of this comes from an owl species that's found in Northern Europe that has 2 color morphs, a gray morph and a brown morph, and they have differential fitness depending on environmental conditions. So when it's snowier, the gray owls are more effective hunters. That results in larger clutch sizes, better success of fledglings, and more gray owls. Versus if conditions are less snowy, the brown owls do better. And as conditions are shifting in Northern Europe, we're actually seeing a shift in the frequency of these color morphs in these populations, which is a direct response to changing conditions. And then the final response that species can have to climate change is to go extinct. And one of the first very clearly tied to climate change extinctions we've been able to note is the bramble key rat, which used to live on an island off Australia, but sea level rise took out its habitat and food sources, and it is no more. So if that's how species can respond to climate change, as a very applied ecologist, as a conservation biologist, I then start to think about, well, what can I do? What can we be doing to facilitate these responses? And I would say we're doing a really good job of facilitating extinction, and I would prefer we do a less good job of that. So I'm going to take that off the list for a moment. Unfortunately for corals, movement isn't an option. Obviously, the adult corals are benthic sessile creatures, but even latitudinal range expansions aren't very possible for corals because they like really stable temperatures around the year. And as you move up in latitude, temperature variation increases. Also, the water gets murkier, and light attenuation means that those symbiotic zooxanthellae aren't getting the sunlight they need to provide the corals with food. So unfortunately for corals, movement is also off the table. And that leaves us with evolutionary adaptation. And about 10 years ago, a bunch of us got together and said, gosh, if that's really what we're looking at, what do we know about evolutionary responses in corals and the timing of that and what we can do to help? So a group of us representing both the nonprofit sector as well as academic researchers got together and launched a research program with the goal of answering 2 questions. Can corals adapt fast enough to keep up with the effects of climate change? And 2, which management actions help or hinder that successful evolutionary adaptation? So our approach was to build a suite of eco-evo models based on Norberg's work from a dozen years ago, and we used these models to explore responses, and we looked at a variety of assumptions related to growth and mortality rates, genetic variance, there isn't a lot of great data about that, dispersal, and we also modeled a variety of sea surface temperature changes through time. And then we're able to test how different approaches to management and conservation perform through time in a system that has both ecological and evolutionary processes working. It's this third piece that I want to focus on today during this talk, but in order to get there, we have to talk a little bit about some of the modeling work so that piece makes sense. So our first model was built by Tim Walsworth. He was then a postdoc in Daniel Schindler's lab at the University of Washington. He's now at Utah State University. And we built a model that had 2 coral types and 1 alga. Modeling coral types rather than species is a pretty common approach because the idea of a species is iffy for corals to start with, and then we have so many. So it's very common to model types. And here we modeled a fast-growing coral that's pretty sensitive to changes in temperature. You can think of that as a staghorn coral. We also modeled a slower growing but more thermally tolerant coral. You can think of that as a mounding coral, like a brain coral. We modeled stepping stone dispersal over a linear temperature gradient. And we simulated management by increasing algal mortality. And this reflects what we can do through conservation and management efforts. For example, by reducing fishing pressure on herbivores, we can actually increase the amount of algal mortality that happens in a system and help corals. It's a less direct mechanism, but improving water quality has much of the same effect. So using this model, here are some of our first results. So this figure here shows genetic variance on the x-axis. Again, we don't have really good data about this, so we wanted to see how that affected our model results. And our y-axis is, for most of the graphs that I'm going to show, our measure of success. So proportion of coral cover is how we think about what we're aiming for. More coral cover, good. And this first graph here shows no dispersal. And then we're modeling out how does the amount of area we've protected, which means managed, affect things. And you can see in this figure that it's not until we have pretty high levels of genetic variance, pretty high levels of protection, that we're starting to see coral cover. But that's with no dispersal. Once you add dispersal, that whole curve shifts to the left. And now even at lower levels of genetic variance and proportion protected, we're starting to see coral cover. So this really underscores the importance of both dispersal and genetic variance in driving some of these results. From there, we then came up with a suite of management scenarios that we were able to use this model to test. The management scenarios in green are all different approaches to protecting diversity. So we evenly spaced our management across that linear array. We randomly distribute it. We used portfolio theory to come up with a set of management units. In the orange and brownie colors, we looked at managing primarily high coral cover or low coral cover areas. And then in blue, we looked at what happens when you prioritize management based on temperature of the reef. And we picked these because there's a very common approach in coral reef ecology. We see it in a number of other systems as well, to protect refugio, which means to protect those places that are currently cool or that are predicted to remain cool through time. So here's our results. And the thing that I want you to take away here is that protecting reefs based on their thermal conditions performs far worse than anything else here. And furthermore, if you look at this then as a function of proportion protected, look at that 30% protected. If you can think about numerous approaches to protecting 30 of whatever system you're talking about by 2030, the 30 by 30 approaches. If you look at that 30% protected, you can see that if you're prioritizing protections based on thermal conditions, you get less coral cover for a unit of effort than if you're protecting using some of these other approaches, particularly the diversity-based approaches. So, to summarize, dispersal protection, genetic variance, all important. Diversity-based approaches appear to perform better, and we appear to get more bang for our buck with those diversity-based approaches. And it looks like exclusively protecting refugia isn't a winning strategy. However, we had to stop and question whether these results were an artifice of this linear temperature gradient we had set up and stepping stone dispersal. So just to make sure that these results really held up, we came up with a next generation model. So Lisa McManus, who was a postdoc in Malin Pinsky's lab at Rutgers, now at the University of Hawaii, led this work. And she built a model that has much more complex dispersal. And you can see this here on the left. We've got an array of temperatures, and we can have connectivity across different temperature gradients. We can vary how regular versus random that connectivity is. And then we can vary how closed or open the system is. And this is much more realistic in the world of coral reefs, where you can have very hot reefs close to very cold reefs. And we're talking, tens of meters distance between them, depending on how close that coral is to the reef edge, the fringe, where there's a lot more flushing versus in a lagoon area where the waters get really warm. So we built out this more complex version of dispersal. We populated this with real-world data from 3 regions, from the Caribbean, the Southwest Pacific, and the Coral Triangle. And then we're able to test how hypothetical marine protected area networks perform in this system through time. So this shows how we're thinking about those hypothetical marine protected area networks. Our regions here are columns, and the rows represent those different approaches to management. So we looked at the existing network. We looked at what happens if we put those marine protected areas, we put that management effort in cold locations. That's the refugia-based approach. What happens if we put them in hot locations? And what happens if we kind of evenly distribute them so that we've got a representative sample of thermal conditions in the marine protected area network? So first results from this was looking at how genetic variance and climate change scenarios interact. So we've got 3 different levels of genetic variance here represented by the colors, with the yellowy color being the highest and the purple color being the lowest. Solid lines are RCP scenario 4.5, so a moderate emissions scenario. Dashed lines are RCP 8.5, so a pretty high emissions scenario. And the takeaway here is that the difference between RCP 4.5 and 8.5 is really notable. If you look at the teal color, which is the intermediate genetic variance, you can see that under 8.5, there's no typical evolutionary rescue curve here. But under RCP 4.5, there is actually hope. So to me, this tells me, 1, genetic variance is still really, really important. And 2, we need to get our emissions under control, and that makes a huge difference for the future, not just of coral reefs, but of many other ecosystems. So the second thing we looked at with this work was the effects of management. And here we're going to talk about our 3 hypothetical approaches to management. So here we've got the blue lines represent our approach to management that puts all our marine protected areas in cold locations. Orange is all our marine protected areas in hot locations. We've got our response metric, coral cover, on the y-axis, and then time running out about 300 years into the future on the x-axis. And the black color here is that representative sample. So applying that management effort, evenly distributing it across the temperatures that exist in each of these regions. And what's really interesting here is the difference between that top row and the bottom row. So the top row is what's happening within the marine protected areas, and the bottom row is what's happening outside the marine protected areas. So when we invest all of our management dollars, if you will, in protecting cold reefs, the MPA network itself does quite well. Outside the MPA network, it's a different story. That actually performs worse. It's the flips for protecting hot reefs, where inside the MPAs, though, hot reefs don't do very well, but outside it does better. And as you might have predicted, that representative strategy that protects a variety of thermal conditions actually does pretty well in both locations. This is starting to get at what's actually going on here, the different roles that these different types of reefs play in this eco-evo system. So coming back to these key conclusions, and now we're really certain that exclusively protecting refugia, those cold reefs, isn't a winning strategy. We know that these hot and cold areas play different roles in evolutionary rescue, and that distributing management efforts across those multiple regimes is most effective. So putting this together, this is a paper that came out a couple of years ago in Nature, Ecology and Evolution. We are starting to understand that these hotter reefs are sources of pre-adapted larvae. Corals in these places are unlikely to survive really long term because the temperature on these reefs is likely to exceed corals' inherent capacity to adapt. But if we're not protecting these reefs now, we're basically not protecting this evolutionary windfall, if you will, of pre-adaptation. In contrast, cold reefs are great sinks for these heat-tolerant larvae. Conditions there are likely to be better over the long term, but if we're only protecting cold reefs, we're failing to protect that evolutionary capacity that already exists in these systems. And of course, intermediate stepping stones are really important between these different areas. So, how does this then translate into conservation and management actions at a variety of scales? So if you'll recall, one of the key results of this research was that management or protection makes a huge difference for coral reefs. One of the things I often heard when I was doing coral reef conservation was, gosh, you know, we're working on this local reef here, and what does it matter? Climate change is going to wipe out everything anyway. And to me, this research really underscores the importance of those local actions. Local here meaning the scale at which an individual or a community is interacting with a resource. And at those scales, we need to reduce local threats. That means combating overfishing, improving water quality, and a variety of other management techniques. And those serve to increase reproduction, settlement, survival. Without that, there's no larvae, there's no genetic exchange, there's no evolution. And it's important at these local scales to also be protecting diversity. The catch is that local scale is too small to fuel evolution. There's just not enough there to really lead to global coral reefs. So we need to be thinking regionally as well. And in this case, that means linking populations to facilitate ecological reorganization, so species shuffling. Reefs of the future are going to look different to reefs of the past, but they can still continue to provide those ecosystem services that we're relying on. We need to help facilitate that shuffling. And importantly, both evolutionary and demographic rescue. And this is the scale at which protecting reefs across a variety of thermal conditions is so important. These narrow approaches that prioritize one type of reef or one environmental condition over another narrow the diversity we need to fuel evolution. So at that regional scale, it's really about that regional planning, marine protected area networks, or the equivalent in terrestrial environments. And then finally, none of this really means much of anything if we don't also simultaneously get our emissions under control. Remember the difference in outcome between those two RCP scenarios. It's really big. So these next decades are going to be critical for global actions to lower the rate and magnitude, both of temperature change and of acidification in our oceans. So with that, a little bit early, but I'll turn it over to questions.
Thank you very much. That was super interesting. And I mean, like at many levels, I mean, conceptual and from the application. Yeah, are there any questions? At the moment, no one's raising their hand, and I think the chat is empty. But what I was wondering, I mean, like at which point are we with like the protection that you mentioned? I mean, what is currently being done? And I mean, how much influence has your organization? I mean, how much, or like the one where you previously worked? I mean, what role is this organization playing, and how much influence can you take on conservation efforts in government and so on?
It's a great question. So around the world, there are numerous government pledges really out there when it comes to marine conservation to protect 30% of marine areas by 2030. And some countries and states and regions are further along in achieving those goals than others. Some have achieved those goals on paper, but not in reality. So there's still a lot of work to be done. And I think as those efforts continue to move forward, it's really advocating to protect the raw material that fuels evolution. Let's not narrow down our options for the future. While we know things will be warmer, we don't know exactly where and when that heat is going to hit. We don't know how heat is going to interact with disease, all sorts of trade-offs. So let's please make sure that we are really protecting a diversity of places and thermal conditions for the future. So at the Coral Reef Alliance, that organization does a variety of local conservation work around the world. So really working to reduce those local stresses. And then the organization, because we're working in a bunch of different locations, is able to both guide and then facilitate collaborative work to do some marine protected area network planning, which happens more at those regional scales, and really advocate for this inclusion of diversity. So kind of a mixed bag on where we are. I think it's similar in terrestrial systems. Some places are achieving their 30 by 30 goals and some lag behind, and some look really good on paper, but perhaps not so good close up.
Great, thanks.
Yeah.
There is one question in the chat.
Ooh.
I try to unmute. I mean, or maybe, yeah, no, I just unmute. Okay.
I can read it out loud if you want.
Normally also, I don't know why. Yeah, maybe we just read it. For some reason, I can unmute.
There's a great
I've asked her.
Oh,
sorry. I asked her to mute, but it looks like it's just text. So
Okay.
So it's a great question, and it's how we increase connectivity between coral populations. Does this involve lab-raised corals, reseeding reefs, etc.? And it's a great question. So there are a few different ways, from less interventionist to more interventionist, to facilitate connectivity. The least is to make sure that marine protected areas are spaced in such a way that natural larval connectivity can happen. So larvae can travel between reefs, and that means spacing, we think, on the order of like 50 to 60 kilometers between those very targeted management units. That's where about 90% of coral larvae settle. So that's the least interventionist end, is thinking about how we locate our marine protected areas to maximize connectivity. You're absolutely right that there are also more interventionist approaches, everything from assisted migration, so movement of corals from one location to another. As you might imagine, that can run into some issues, particularly if movements are happening over really big scales. Governments, for good reason, have some concerns about that. We don't want to be moving disease with corals or introducing invasive species accidentally. But there are a variety of kind of assisted migration options out there. And you asked about, raising corals in labs and reseeding them. Yes, there's a number of groups out there doing that work. Right now, it's very hard to get to scale. It's expensive. And it's unclear how well these corals are actually surviving, because if they're put in a location where effective management isn't happening, you're just putting those corals back out where they died in the first place. So as far as I can tell, some of those most effective versions of replanting corals are doing, about 10,000 corals a year. We estimate that some of these natural reefs have millions of corals. So there's a real scaling issue here. Yes, there's also work done or starting to look into reseeding. It's quite complicated. And again, these more interventionist approaches don't mean anything. They're not going to do anything, have any effect, unless we're reducing those local stresses in those key locations. Otherwise, we're just putting corals back out where they died in the first place. So again, I'll just emphasize the need for actions at multiple scales. There's no one solution here. It's going to take a bunch of work. I like to say there's no diet pill for coral conservation, right? We actually have to get out there and do the work. We need to be working at local scales. We need to be doing regional planning, and we need to be thinking about global effects as well.
Okay, so if there aren't any more questions, I don't see any, then yeah, thank you very much. And let's move on to our second speaker in this session. So the next talk is by Austin Bird from Imperial College London. And there we are switching now to gene drive systems with applications to combat malaria.
All right, hello, everybody, you can hear me fine?
Yes.
Great, okay, thanks. Well, thanks very much for the invitation to give this presentation. So prospects for genetic biocontrol. Genetic biocontrol is something that's been practiced for decades now, in particular the use of the sterile insect technique. So that's where you suppress or eliminate a population by inundating it with sterile males. And this has been used for insects in traditionally, the sterilization is done by irradiation, but it can also be done by introducing a Wolbachia bacterium into the males so that they have it, and if that bacterium is not in the target population, the females, then that can cause matings to be unproductive. Or there's genetic modification, transgenic approach, the release of insects with dominant lethals. There's been some good success of these approaches against some agricultural pests, such as the screwworm has been eliminated from North America decades ago. Medfly is currently controlled quite successfully using the release of sterile males in various parts of the world. Tsetse fly was eliminated from Zanzibar by releasing sterile males. So there's good successes in some cases, but it's impracticable, this approach, when the target population is very large or if the species is difficult to rear. So a paradigmatic example of that, for example, is Anopheles gambiae mosquitoes in Africa, which is the main vector for malaria. Malaria is causing hundreds of millions of infections every year, hundreds of thousands of deaths. And there's just way too many mosquitoes over too much space. It's a rural disease, so across rural Africa, to possibly think about inundating this population with sterile males. Also for decades, at least since the 1960s, people have wondered about whether using self-motivated the process of gene drive, as discovered in various sorts of selfish genetic elements, whether that might be useful for controlling pests. The classical premolecular genetics was not sufficient that anything applied came from this. But with recent advances in molecular and synthetic biology, this sort of approach, motivated by natural examples of synthetic selfish genetic elements, now looks more promising. And there's lots of discussion and work, research, investigating the possibilities here. There's three types of target population that people are considering: disease vectors, or harmful invasive species, or agricultural pests. And the different taxa, of course, are going to require different sorts of solutions, but there's just lots of investigations happening at the moment for the potential of using genetic approaches. So what is gene drive? Let's make sure we're understanding what that is. So gene drive is a process of preferential inheritance. That's one part of it. So that if you've got a plus and a minus gamete coming together to make a heterozygote, instead of that heterozygote making 50-50, passing on the two genes 50-50, if there's a bias there, so it's 70-30 or 95-5, then you've got an increase in the frequency of the plus allele, in this case, just due to the pattern of gene transmission from one generation to the next. That means, all else being equal, that gene will increase in frequency in the population, and that can be relatively rapid, so that if it's a, for example, 70-30 split, then you can go, all else being equal, go from 1% to 99% in 20 or so generations. And if we're talking about mosquitoes, think about a generation every month or so. Okay, so gene drive has evolved many different times, naturally, in diverse ways. And it's a process that we're beginning to know more and more about the molecular biology of it and how to mimic it. Quite attractively, it can lead to the spread of genes that are, even if they cause that gene causes some harm to the individuals carrying it. So normally, of course, genes can only spread if they increase the survival or reproduction of an individual. But here it's showing drive, it can spread through a population even if it's causing some harm to the individuals carrying it. And so that makes it an attractive process to try and exploit for controlling pests and vector-borne diseases. In the field, there's sort of two basic strategies that are talked about for how you might use gene drive, at least in the context of vector-borne diseases like malaria. One is to suppress the population. So you release your gene drive, it spreads naturally through a population, increases in frequency, and as it does so, the numbers come down, and it eventually crashes the population, so you're suppressing it. Or alternatively, there are ideas and strategies about population replacement or modification, in which the gene drive spreads to the population, numbers stay more or less intact, but you're changing it to be less harmful, such as being unable to transmit malaria. In principle, the two approaches can be combined, and we mostly work on suppression, but there's other groups that are working on replacement, but I'll be talking about suppression mostly today. So what's the mechanism by which you can get into more than 50% of the gametes? So as I mentioned, many different types of genes have evolved mechanisms for doing this. The mechanism that has been showing the most promise thus far in developing synthetic versions of gene drives uses the so-called homing reaction, in which you've got, if you're a heterozygous individual for one of these genes, it contrives to get itself copied across to the homologous chromosome, so a heterozygote turns into a homozygote. And so instead of getting to 50% of the progeny, it gets to all of them. It does that by, if you look in the middle there, you've got a gene, a homing endonuclease gene that makes an RNA and an endonuclease that goes back to the nucleus and recognizes a specific sequence and cuts that sequence. The gene is in the middle of its own recognition sequence, and so the presence of the gene protects the chromosome from being cut, and it's only the chromosome without the gene that ends up being cut. Now you've got a broken chromosome, and that then is typically repaired using, at least in some species, by using the homologous chromosome as a template for repair. And so after the repair process, you've got the gene on both chromosomes rather than just one. So this is a natural process that is used by homing endonuclease genes as found in many microbes, yeast, and other fungi and algae, etc. These are genes which show apparently no benefit to the organism and just spread because of this non-Mendelian inheritance, this drive. So how might we use this? We can imagine finding a target gene in our target species, defining a recognition site within it, and then designing a nuclease that will recognize that recognition site, the 20 base pairs, let's say, within our target gene. We insert a gene encoding that nuclease into the middle of its own recognition sequence. So that then, again, protects the chromosome it's on from being cut, but you've whacked it in the middle of a target gene, and so that's going to disrupt the target gene. And if you put a germline promoter controlling that nuclease gene, such that it's turned on when the organism is making its sperm or its eggs. And so then, if you've got a heterozygous individual, it will, when it's making its gametes, that nuclease will be turned on. It'll cut the target gene, get copied across in the repair process, get inherited by more than 50% of the gametes, and spread through the population. And you end up being able to catalyze a population-wide knockout of your target gene. The impact of that, of course, is going to depend upon what sort of target gene you go after. And for this, we've been doing this for some years now. And tried many different nucleases, architectures, and we're now using CRISPR-based ones, because they're so easy to work with, relatively speaking. So the molecular biology work I'm going to talk about, show you today, is being done in the lab of Andrea Crisanti, also at Imperial College. And one of the genes we've been looking at is, so we're, as I said, mentioned in population suppression, so interested in targeting genes that are involved in female fertility, such that the homozygote knockouts are female sterile. So I've been looking at this gene in particular, double sex, and it's a gene involved in the sex-determining pathway of Anopheles gambiae and most other insects. There's an exon in it that is only expressed in females. And if you look at so there's differentially spliced to this gene in males and females. And we've been in particular targeting a recognition sequence that spans the intron exon boundary of that female-specific exon. And you can see not surprisingly, it's well conserved among different species. So it's probably functionally important, and the conservation level suggests that's actually true. When we insert a gene into this region and knock it out, we find out that homozygous females are indeed sterile. And as a bonus, they also can't blood feed because their mouth parts are disrupted. But there's no obvious effect of the homozygous knockout on males. All right, so we put in a CRISPR-based nuclease into the middle of this sequence, targeting the natural recognition sequence, and looked at the inheritance out of male and female heterozygotes for this CRISPR-based construct, and found 97% transmission rate. So very high transmission rate, far higher than the Mendelian 50%. So that's very promising. And then we've also shown that you as it would be expected, we got spread through a population, through replicate populations. So the red and the blue lines there show in the top graph the frequency of individuals from the construct increasing over time. So you just see the population with, in this case, 25% frequency of individuals, and then just let the population run over time. And the gene of its own accord, because of this homing reaction, increases in frequency. And then on the bottom graph, the egg output is going down, and the population crashed until they went to 0, 8 or 12 generations after starting. And we've also done this, so those are just in small, relatively small cages. We've also done it in larger cages that are better able to mimic at least the physical environment of the mosquitoes. They're large enough that we can allow a place for the males to swarm. So there's a somewhat more natural mating environment. And again, we saw the same sort of thing, that the gene driver is able to spread in the population over time, replacing the wild type. And as it does so, the egg output of the populations decreases until eventually they're eliminated. And in different populations, in different experiments, we've shown population crash more than 10 times in different experiments. So it's a repeatable experiment, or a repeatable result in the lab anyway. Okay, so that's some of where we are, or where we are partly with the molecular work. We also want to know, this is promising results. We want to know, well, what would this actually mean for malaria transmission? This is what we're trying to, of course, be affecting. So this is some recent modeling work we've been doing to try to investigate that question. Well, what kind of impact can we hope to have? And so we focused in particular on West Africa, and took 16 little 1 degree by 1 degree cells within that, chosen in order to represent a diversity of ecologies and malaria prevalences and the like. So each cell is, as I say, 1 degree by 1 degree. Penny, Ace, Adrian, and John, the ones leading this modeling. And so what was simulated was to release just 1,000 males of each species. So there's different species as vectors in here. There's Gambiae, there's a Colucii and Arabiensis. These are 3 closely related species, sibling species of the same species complex, plus a more distantly related Anopheles fenestus. So we simulated release of 1,000 males in each of the species, in each of the 50 just randomly selected settlements in each one of those 1 by 1 cells. So that's somewhere between 1 and 30% of the settlements. 50 settlements is, and that's sort of 0.04 to 4% of the estimated number of mosquitoes that pre-existing in that cell before release. And this plot then shows for each of the 16 areas the relative population size or the degree of population suppression 3, 6, or 12 years after the initial releases. So if they had no effect, you'd be up there at 1, 0 suppression. You see all the bars are well below that. And for the most part, most of the areas that we're getting more than 90% suppression of the populations. So it's really highly effective. This relatively small intervention can be highly effective. And that efficiency is then due to the leveraging, because they were leveraging normal biological processes of DNA repair, of the reproduction of the mosquitoes, of the dispersal of the mosquitoes. So by dispersing, the gene drive gets from one settlement to another. And so it's leveraging these normal biological processes to make something quite efficient. In that way, it's in some ways analogous to vaccines, which of course are very efficient by leveraging the adaptive immune system we have. And that efficiency, of course, is very important in the context of a very, very large geography without a lot of resources. Okay, then we've also looked at the effect of this on malaria prevalence. And so this is a complicated graph showing for each of the 16 areas, what are the effect of releasing into one species, another species, a third species, or all of them combined. And you can see, not surprisingly, that the transmission, you're going to have an effect if you release it to species that are the vectors in that particular location. And if you're not targeting the important vectors in that location, then you don't have much of an effect on transmission. But if you do manage to target those species, then the impact can be quite substantial. Also shown by modeling will show that gene drives can act positively in combination with vaccines, the new vaccines that are being developed, and with the new bed nets that are being developed. So it works positively with these other interventions in reducing clinical cases. Just as an aside, I'll mention these models, of course, need parameters and values to go into them. And we're doing what we can to analyze population genomic data to infer some of these parameter values. So we kind of need things like population sizes, movement patterns, and something more information on what's happening in the dry season. Ecology would be much desired. Sort of two sources of information we're using here are SNPs in the AG1000 database and the positively selected mutations as exemplified by insecticide resistance. So that's data being generated largely out of the Sanger Center here in the UK as part of the Malaria Gen program. And so the sorts of things we're looking at are temporal fluctuations in SNP frequencies over time, lengths of shared haplotypes, and geographical distribution of insecticide resistance. This plot is a very nice data from that Sanger Center collaboration, looking at how the geographical spread of different origins of the same insecticide resistance mutation in different populations. And you can see, as you might imagine, there must be data in there, information in there on population sizes and on movement rates, and we're trying to find out ways to extract that information. Okay, just to emphasize, we as beginning started, of course, thinking about could we ever get a synthetic gene drive to work, to operate in the lab? But there's a lot more to eventual success for this kind of project than just technical success. So yes, we do need to know, can we get something to work? And so we're doing cage studies and we're doing a lot to figure about how to prevent resistance evolving. But there's also big questions about, is such a thing acceptable? And acceptable both to regulators and to the broader public and stakeholders at large. And for that, of course, we're having to do risk and impact assessments. providing information to people and addressing their concerns to the extent possible in order to facilitate informed decisions being made. And then there's a question of whether this sort of new technology can be delivered. And so that's an issue of capacity building, building facilities, training personnel. And a large part of what we're doing here is to drive this, both the acceptance and the capacity building issue, is doing studies with non-gene drive genetically modified mosquitoes. So we have this sort of step-by-step phased approach in which we don't think going to Africa with a gene drive mosquito as the very first thing is the right way to do it. Instead, we're building up to that step by step. So we have our first strain was a dominant sterile male strain. So it's just no offspring. The males don't produce any offspring. You release the males, there's not going to be any persistence of the gene. So that strain we've imported into 2 countries in Africa, Burkina Faso and Mali, and integrated into the local genetic backgrounds. And we've done a single small-scale release in Burkina Faso and monitored the disappearance over a single generation of that strain. We've also got a paternal male bias line, in which the males are fertile, so they produce progeny, but 90 to 95% of those progeny are male. The genes responsible for this are autosomal, so it's not driving. So it's expected to, if we release it into population, it'll persist because the males are fertile, but gradually disappear from the population over time. That line has been imported into 2 countries, into Burkina Faso and Uganda, and we're planning for lab studies, and we're planning conditional on approval to do some releases in Burkina Faso to look at the dynamics over multiple generations. And then if all goes well, then the intention would be to move to working with doing small-scale trials with a gene drive line. There's a lot of stakeholder engagement involved in this. So all those, and that happening at a variety of levels. So the WHO, the African Union, OECD, EFSA, CBD, and the national academies in the US have all produced documents on gene drive, all shown a great deal of interest in this sort of technology and the possibilities of it and of the issues arising from it. So we've been involved with that. And then, of course, there's at the other extreme, stakeholder engagement in the particular villages where our African collaborators are working. So in the insectaries in there, we've had open days, so inviting people into the insectaries to show them what's happening. We've done that both in Africa and our labs here in Europe. And also in the villages where our medical entomologist colleagues are working, there have been efforts to explain what we're doing and what the technology is all about, including a theater forum to explain the sterile male strain, the release of that there in Burkina Faso, with country-specific brochures and graphical posters. Collaborated with linguists to make a glossary of terms in local languages, made short explanatory videos, and the listening to concerns to incorporate into our risk assessments about what could go wrong. The primary goal here, of course, being to facilitate, as I mentioned, informed decision-making. So just to reiterate that there's much more than technical success that's required. We also need to ensure that this sort of technology is acceptable and that it can be delivered appropriately. Just very briefly, just in a couple of minutes, I'll mention that this gene drive approach that I've discussed thus far is going to spread throughout a species range if there's almost any kind of gene flow. And while that may be appropriate for Anopheles gambiae, which is basically harmful everywhere it exists, there's a lot of other potential use cases where you don't really want that to happen. You don't want to have an impact much beyond the release area. So an invasive species, for example, you want to control it where it's invasive, but not where it came from, etc. And there's also a lot of work being done on approaches for that sort of problem. So sorts of things we've been interested in are looking somewhere between sterile males, which disappear immediately after you release them, gene drive, which is not localized because the causative factor spreads through a population and therefore geographically. So in between those two things of disappearing and spreading is something that just persists at whatever frequency you introduce it. And so we've been exploring different ways of potentially doing that. One of them being to have a gene on a Y chromosome that makes mutations on an X chromosome. That means the daughters of the male, assuming the male is heterogametic here, so they're dominant. That's a dominant lethal or dominant sterile. So the daughters inheriting that X don't reproduce. But that doesn't affect the Y. And so at least under ideal circumstances, that modified Y chromosome would stay at whatever frequency you put it in at. So if you put it in at 70%, then every generation, 70% of the females die or are sterilized, but that doesn't affect the frequency of the Y. So that sort of thing could be 10 or 100 times more efficient than sterile insects, but still remain localized. And that's just one example of many things that are being developed in different labs and groups and taxa around the world. So it's quite a thriving field at the moment, this application of synthetic biology to biological control. So, in summary, great potential for genetic biocontrol to help tackle some of the outstanding challenges we face. And population geneticists and evolutionary biologists really should be at the heart of these efforts, both in the strategy development, what sort of approach is best for a particular use case, and for population genetic analyses of your target population, for tactical models, and for helping decide when the molecular biology is good enough. Just to acknowledge, all this is done under the auspices of Target Malaria, which is a not-for-profit research consortium developing this sorts of technology, diverse expertise in Africa, Europe, North America. Our African PIs are shown there on the right, and we get core funding from the Gates Foundation and the Open Philanthropy Project. Thank you very much.
Thank you. That was really great. I mean, it's what I find also very fascinating, how this shows how people cooperate and collaborate. So it's very nice. Yeah, so are there any questions? Then please just raise your hand. I already see one.
Hi. This question is related to the community engagement, stakeholder engagement in local villages and so on aspect.
Sure.
Do you have any examples of times when a planned trial was modified because of some community concerns or feedback?
Well, so certainly, where we do, the precise location of where we do a release in a village and where we're going to do the collection and how we're going to do the collections and the monitoring. Yeah, that's done in consultation or collaboration with the village. The first question we ask is, how would the village like to communicate its approval or disapproval of what we're doing? And because we have to listen to them in terms of how their governance system works, and so that sort of level. We're not asking them to modify, which guide RNA we should be using in our molecular constructs, but the practicalities of what we do in the field, yes.
Right, thanks.
And there's another one.
Hi, yes, I have kind of a general question about the application versus kind of the basic science. So I've been working on sex ratio chromosomes, and I'm wondering, is the basic biology of natural gene drive systems particularly useful, or is it really about just applying what's already known about synthetic gene drive systems?
Well, the natural ones, naturally occurring homing endonuclease genes were really useful in giving us the idea that we could use nucleases to make a synthetic gene drive. So they were kind of inspirational in that regard. So in insects, as you mentioned, there's naturally occurring sex chromosome drives. And I think it's unclear to what extent we can learn from those, yeah, unclear. I think it would certainly be nice if we knew more about how mechanistically those worked, because that might, again, give us ideas about how to make synthetic sex chromosome drive, which has some nice properties. We've tried doing that. The gene that we've got on the autosome that makes a skewed sex ratio. If we could get that active on the Y, that would be great. But so far, that's not worked. So mechanistically, I think that could be very useful. Yeah.
Would it be fair to say that the future of the field is much more about the application of the existing technologies then, rather than trying to develop new technologies? Or
oh no, it's a very young field. Would not want to say we've discovered everything useful about and it's just a matter of application. No, and new ideas are coming out, seems like every month. So I think that's all to play for still.
I have a very brief question, which is not scientific, but I mean, this seems like there are really a lot of people collaborating together. There's this consortium. I mean, how does one get into such a collaboration? I mean, is it one of the things that just happen, or is it something that you actively seeked?
Well, our primary funder is the Gates Foundation, and there's nothing like a joint funding stream to build a collaboration. So, and they've been very generous with us over the many years now. And, obviously it's grown over time to get to this size, but because we didn't start anywhere near this big. But yeah, I think it's having a funding stream like that has just made everybody want to work together more than is perhaps easier in other circumstances. And they're quite happy to fund people as long as they fit the project. They're quite happy to fund people anywhere in the world. So you can get collaborations internationally quite naturally coming from that. So they've been very, very good funders.
Great, thanks. Okay, then I think there are no more questions, and we are also already a little bit over time. So then thanks again, and let's move to the last speaker of the symposium, which is Ayari Fuentes Hernandez. So she's from Mexico, and she's going to talk about the evolution of antibiotic resistance.
Thank you very much. Thank you very much, Hildegard, for inviting me. And let me. Can you see my talk?
Yes, it's working perfectly.
Okay, cool. Well, thank you very much. As Hildegard said, I'm Ayari Fuentes Hernandez. I'm from the Center for Genomic Sciences at UNAM in Cuernavaca, in Mexico. And today I want to talk a little bit about the work that we are doing in my lab about evolutionary history and selection, particularly using experimental evolution in microbes. So first, I wanted to talk to you about why it's important to understand what is happening with antibiotic resistance. And the thing is, antibiotic resistance and I think clean water are the two things that have changed our lives, like human beings, a lot. And at least with this data in the mortality in the US, before we got antibiotics in the clinics, almost all almost all the people that die actually were because of infectious diseases. Almost 60% of the people that die. But then we got antibiotics, and for example, in this data in the 2010, just around 10%, well, 16% of the people who die actually now die for infectious diseases. So why is this a problem if actually the rate at which people is dying decreased a lot? And the problem is that, and we know this since the antibiotics were introduced in the clinic, we found resistance very, very fast to any antibiotic that we have been using. So, for example, in penicillin, we found the first clones of resistant to penicillin, like I don't know, like some years before the introduction of the antibiotics. So this wasn't a problem because there were a lot of antibiotics introduced in the clinic. So we just keep using one. We found that this antibiotic was not working anymore, and then we just introduced another one. But the thing is, from the 1980s, it has been a void of the introduction of new antibiotics into the clinics. So now we are in a very, very big problem. And from the point of view of basic science, I think that the thing that we can help to fix this problem, which is going to be a very big problem in the next years, is trying to understand what is happening and how populations are becoming resistant to antibiotics, and moreover, try to propose new treatments with the antibiotics that we already have. So in order to, well, the thing is, this antibiotic resistance is a very difficult problem because it's a multi-scale problem. It's a problem with very different layers. We have very spatial scale. Well, we have very spatial scales, and we have also temporal scales. So in order to do that, the work that I'm going to talk about now was a collaborative project with Rafael Peña-Miller, Sandra Cisneros Mayoral, Lucia Graña, y Deyanira Perez, which at the moment of this work, we were all at CCG at UNAM in Mexico. And what we were interested in is actually, given that one bacterial population, well, if you have a fitness landscape, and the peak of the fitness landscapes are actually the amount of resistance that a population can get. If we were interested if the way at which the population is getting this resistance, is going to affect the outcome of the population. You know? So in this, if you see this graph, the peak of the population is when a population is resistant 10 times to some antibiotic, and I'm drawing there some special way that you can get to there. But there are another peaks that are at the same level that if you just measure it phenotypically, for example, those two peaks we cannot distinguish between them because they are equally resistant. But then, is it going to be relevant if you get to one peak or to the other? And of course, the ways are going to be very different. And in that sense, we are interested in memory and in evolutionary history. In order to tackle this question, we are going to use adaptive evolution, laboratory evolution, experimental evolution, which is we get a population of bacteria. In this case, we are going to use Escherichia coli. And we are going to have a clonal population from this whole population, and we are going to transfer this population to liquid fridge media, and we are going to let them grow for some time. And then we are going to have a proportion of this population, and we are going to put it into another flask, and we are going to do that for several days in order to actually understand how this population is going to become more fit to this particular environment, of course. And we are going to see kind of evolution in real time. But then in the sense, when we are using actually antibiotics, we can do very cool experiments with this kind of setup. We can use just constant environments. We can put always the same amount of antibiotic. We can use sequential treatments. For example, we can put one day on one antibiotic, on one day the other. We can switch between antibiotics or between selective pressures. We can switch between putting antibiotic one day and removing it the next day, or we can do ramps of antibiotics, which is the one that we are going to use in this project. And a ramp of antibiotic is just each day that you change from one flask to the other, you put a little bit of antibiotic and you start increasing the amount of antibiotic within time. So we can actually study how this population is going to become resistant within these different these different treatments. So Another thing about the working with microbes is that we can freeze everything. We can put it in ultra freezers, and then we can go and we can do whole genome sequencing within every time point that we want, and we can also took these clones and actually do more experiments to see what is happening. So when we start this project, we were trying to do actually a mathematical model to try to understand how this is happening in a dynamic way, how this experiment can be modeled in a dynamic way. So using just a very, very simple ODE model, just a 3-allele model, 1 locus, we wanted to see exactly the dynamic of a ramp of antibiotics. So we started with susceptible bacteria, and these bacteria in the model can become resistant in 2 ways. Just by a point mutation, that is, this mutation is going to occur just by a probability. And these susceptible bacteria can become very strong, resistant at a very high cost, or mild resistant at a lower cost. So in that sense, this is how it works. In the absence of antibiotic, the susceptible bacteria is going to be the one that has the higher bacterial density, is the one that are going to grow better. But as soon as you start putting antibiotic, of course, it's the one that is going to die first. And the cost is going to be measured just in the absence of antibiotic. So when we have this model that works perfectly, and it's a ideal model, what we did is just a ramp of antibiotic, and we decided to first do a mild selective one. And in here we were actually asking about how was the population structure of this very high of this population that was resistant to some antibiotic. So we did this ramp. So as you can see in the X, Y, and the X-axis, we have time, and in the Y-axis, we have relative frequency. So at the beginning, we have all wild types, and then as soon as we start actually putting more antibiotic, it start becoming a little bit resistant. But then if we now use another way to get it exactly into the same point, and we use a strong selection, what we realize is the profile, the population profile is different, in the sense that we have more bacteria that is very, very resistant than in the mild selection. And the other, of course, the other very big difference is that the rate of adaptation is higher in the strong selection. And by rate of adaptation, I mean the time at which this population is getting resistant. And remember that this population at the end of this experiment is exactly the same resistant. So it's 10 times more resistant than the initial one. So in the terms of phenotypes, we cannot distinguish between the two. So once that we have these 2 populations and the population structure is different, what we wanted to test is what if we remove the selective pressure. So we continue this simulation, but now with drug-free environment. And of course, we were hoping that in drug-free environment, as these mutations were costly in the model, we were going to lose these mutations because they don't need it anymore. But then if you realize for the mild selection, the population is not completely naive as for the strong selection. I mean, the population became completely susceptible for the strong selection, but not for the mild selection. And this is interesting because what if we put again some antibiotic? So if we introduce antibiotic, what is happening is that for the strong selection, the population is kind of the same as the very beginning, at the phase 1. So they have to start again getting all the resistance. But for the mild selection, as the population has already resistance in there, it's not completely naive. So the rate of adaptation, the time at which they are becoming resistant, is much, much faster than the phase 1. So this is kind of, well, this was kind of weird because this means that mild selection actually have more memory. I'm going to call it that strong selection. So the resistance is kind of more stable in mild selective environments than in the strong selective environments. So we were really interested in this. This was a kind of a toy model, of course. So then we decided to went into the lab and to test this hypothesis, if this is really true. So we decided to do the evolutionary experiments. We use Escherichia coli. We use ampicillin as the antibiotic that we were going to test. And what we did is every time we decided to do a dose response curve, which means that we are going to have 22 different drug concentrations, and the clonal population we're going to grow in those 22 drug concentrations. So in there, we were going to determine the MIC, which is a minimal inhibitory concentration, which is the concentration at which everything has died. So we cannot measure any cell in the environment. And then for the strong selection, we are going to take the concentration just before that, which is going to be the concentration that kills almost 90% of the population. And for the mild selection, we were going to choose the concentration that's was going to kill the 50% of the concentration. So these were two different regimes, and we were going to do it until all the populations were 10 times more resistant to the what they want. So we did this, and this is the result. And for the strong selection, well, in the picture we have generations in the y-axis and MIC in the y-axis. So MIC is the amount of resistance, and as you can see, the strong selection get resistant really fast, like really fast as the model. But for the mild selection, it's more noisy, and it take almost 3 times more generations to get in there. In terms of time, the strong selection actually was really fast. It was around 7 days, and for the other one, it was around between 22 and 25 days. So the difference was really high. So we got that the rate of adaptation actually was correlated with the selective pressure, as exactly what we were expected. But then what happened now for the second part of the experiment? Like, we took these two populations that if you measure the level of resistance, is exactly the same. But then we are going to remove the antibiotic in the environment. We did that for 8 days. And what we realized is that for both treatments, actually, they become susceptible. Like, what we were measuring, again, dose response each day to measure resistance. And as you can see, for the mild selective regime, which is the blue one, the resistance actually is, take longer time to decrease. It take almost 10 generations to decrease. And the decrease is really, really, well, it's decreasing in a really slow rate. Sorry. And then for the other one, for the mild selective pressure, for the strong selective pressure, the decrease was really fast. Like, within 1 or 2 generations, all the population was already susceptible. And this kind of makes sense in this. We have the hypothesis that if the population was getting resistant, so that fast was because the mutations were really punctual, and maybe they were really costly. So if they were really costly, when you remove the selective pressure, they were going to lose it very fast, which is kind of the case. But the other interesting part is, as the model, actually, the mild selective pressure is not as susceptible as the strong selective pressure. The mild selective pressure is not getting until through the lowest level as the wild type, as the high selective pressure. So now we were interested in what happened when we reintroduce again the antibiotic. We reintroduced the antibiotic exactly in the same way that we did it before. For the strong selective pressure, we did exactly the same ramp. And for the mild selective pressure, we did exactly the same ramp. And this was really interesting because, again, in the y-axis we have MIC, in the x-axis we have generations, and in here we are comparing the two phases, the first phase and the second phase for the two different regimes. And for the phase one, which are the solid lines, the only one that we can actually differentiate is the phase one for the mild selective pressure. The other three are kind of the same. And the phase three actually for the other two regimes, for both regimes, for mild selective pressure and strong selective pressure, were even higher in the kind of same generations. So it was very interesting again to see, particularly the phase three in the mild selective pressure, to get resistant so fast and to be very, very resistant, more resistant even than the strong selection for the same amount of generations. And then if we measure the rate of adaptation, for the strong selection, the rate of adaptation actually is constant for both phases. It's not accelerating. So it's kind of, it doesn't have memory that already saw the same antibiotic. But for the mild selective pressure, the rate of adaptation for phase three is very, well, it changes a lot, it's higher, but it's even higher than the strong selection. So this kind of memory that it has from already seeing the antibiotic in the phase one, it's getting a very good advantage from getting the resistant to the same antibiotic. So then we wanted to measure the cost of resistance at the end of each phase. So in here we are having this relative fitness. If it is above the line, it means that it has a positive cost. It means that it already compensates the cost. And if it is under the line, it says that it has a cost of resistance in comparison with the wild type. So for the mild resistant, actually the fitness cost remains quite constant. Is costly to be in that phase. But if you see for the resistant mutations, particularly in phase two, they rapidly compensate the cost and they even grow better without the antibiotic that the wild type, which is exactly what kind of our hypothesis at that point. And then in here we are just seeing like all the experiment all together. And we are seeing the mild selection, the strong selection, the different replicas. And then if you can see the difference within time, it's just the first part of the experiment, like for the mild selection. The other part is kind of the same. And what we did the next is we took those points, the dots, the color dots, and we performed a whole genome sequencing to see if the genetic profile were different or not. And then what we realized, these are the results of the whole genetic sequencing. We did it for the whole population. So in the y-axis we have a mutation frequency for the population, and we are dividing it for phases. We performed this whole genome sequencing at the end of each phase. So what was very interesting is that the amount of mutations that we actually have for strong selection is quite few. For the first phase, there were just 3 mutations that make the whole population become really resistant just in 7 days. And then as soon as we remove the antibiotics, we actually see the decrease of this percentage of the decrease of the coverage of the mutations in the population. So they were actually losing the mutations. And then when we reintroduce the antibiotic, we see a bit more of mutations, but we actually see again another mutations. We don't see the same mutations in phase 1 and in phase 3. And now for mild selection, it was really interesting because we saw more mutations, which is kind of, this was not that surprising because at the end of the day, like the first phase of mild selection has more time, so you have more accumulation of mutations. But we have several mutations. We have several mutations and 100% of the population. We were some mutations that were at the 50 or 70%. But then what that was really interesting is that those mutations that were at some part of the population at the end of phase 2 were already fixed in the population. So they were at the 100% of the population. Even though at the end of phase 2, remember that this population were already more susceptible to the antibiotics, they didn't lose the mutations that gained in the phase 1. And then when we reintroduce the antibiotics in phase 3, we find some more mutations, but not a lot. So the thing is, this rapid gain of the adaptation that has in phase 3, it has to do with the mutations that already have, not new mutations. So this is kind of that. This is why I keep saying that this has like kind of memory. No, this is a memory case of the mild selection actually put memory in the population. And we were interested in this because this actually can affect how the treatment outcome, isn't it? If we are just measuring phenotypes, we cannot differentiate between these two, but these two are very different populations. And then when we realize this, we say like, well, if they are genetically different, maybe the profile actually of the cross-resistant is going to be different. So kind of ongoing work, and I'm sorry, like this is the first preliminary data, but what we did is we took samples from the end of phase 2 from selective pressure from SS and from MS from the two different treatments, and now we are trying to measure cross-resistant with different antibiotics. So we use amoxicillin, ceftazidime, gentamicin, piperacillin, ciprofloxacin, and doxycycline. And we were hoping that actually the strong selection and the mild selection were really different in the sense of cross-resistant and cross-sensitivity. We are kind of puzzled of these results, but we do have a lot of cross-resistance, which is the thing that we kind of hope. Mild selective pressure have lots of cross-resistance, particularly in antibiotics that are not beta-lactams, as ampicillin, which is the antibiotic that we are using. But interestingly, in both cases, we have cross-resistance for beta-lactams, which is really, well, at the beginning it puzzled me, but then it's not that, well, it's not that weird. So maybe we can actually try to use, like, if you evolve a population to one beta-lactam and this is becoming, this is making more sensitive to other beta-lactams, maybe we can actually do treatments with just beta-lactams to play with this cross-sensitivity. But then this is all for the this is all preliminary data. We have to start. We have to do the whole genome sequencing now in this experiment. But well, I think this is relevant, and actually understanding how different selections make different genotypic profiles is really relevant to understand antibiotic resistance. So just to conclude, we know that the rate of adaptation is proportional to the strength of the selective pressure. We know that, and this is not very surprising, but the strength of selection actually shapes mutational spectra, and this makes that mild selection favors the stability of resistant mutation. And I think we have to also start looking for mild selection, because it's really relevant now if we see that we actually found lots of contamination by antibiotics in water and in soil. So this contamination can be very mild it can be selection in a mild way, but actually it can be shaping like some populations. And evolutionary history and strength of selection is going to determine the rate of resistant adaptation, and when this could be maybe relevant for cross-resistant, and maybe this could help us to design better treatments. So thank you very much. Thank you very much for the funding bodies. And this is Cuernavaca. This is a nice place. If you have the opportunity, you should visit. And well, this is the group. And thanks also to my group and to you for listening to me.
Thank you very much. I mean, that's very interesting. I especially like this sort of last results that you showed about the cross resistance and cross sensitivity.
Yeah.
But has anyone? Yeah, I see there is a question already. Is it?
Yeah.
Yeah, worked.
Hey, Ayari, great talk, by the way. My area of research is also mutations. So I have a couple of questions for you.
Yes.
You showed the mutations that have occurred both in mild selection and strong selection, but have you assessed their effect on the proteins? Like, have you assessed like whether they are like non-synonymous mutation, whether they are like not as in like changing the amino acid code, or like whether they are not changing the amino acid code, because we know that the code is degenerate. And based on that, have you assessed whether what kind of like selection is happening, like purifying selection or positive selection, or if there is a directional selection, something moving in one direction, a trait or a
Well,
fitness?
That's a great question. We do have which one are synonymous or not synonymous. In here, actually, this is the things that I show is quite narrow because they are just SNPs. We did something more, but I'm not presenting it. But no, we haven't assessed that part. And every time I give this talk, someone asks me that. So we should go again into the data and actually see that. But no, we haven't done that. Thank you.
Right. Okay.
I can hear you.
Oh, sorry. Yeah, I just got a mistake. I'm so sorry. So I said there's a question in the Q&A. Can you see it? Also, like I read it again. So it says, thanks for a nice talk. In phase 2 under strong selection, can you distinguish between compensatory evolution and replacement of resistant genotypes by sensitive but more fit genotypes?
Sorry, can you repeat it?
Can you see it in the Q&A?
Oh, yes. Yes,
it?
okay. In phase 2, under strong selection, can you distinguish between compensatory evolution and replacement of resistant genotype by sensitive? No, we cannot do that the way we did the way we did the whole genome sequences, because we just put all the population together and we just in a very high coverture, and then we did that. So no, sorry. We should do that, but no, we haven't done that.
Okay, it says thank you.
Yeah.
Okay, I do not see any more raised hands, so it seems there are no more questions. So yeah, thank you again for your talk, and thanks all the speakers. I think that was a really super interesting symposium, and I enjoyed it very much. And yeah, now I think that's